Welcome, and thank you for joining us today for this Lab Roots webinar. Our topic today is leveraging the plant genome to increase crop yield and quality. We have three outstanding speakers for you today. Gawain Bennett from the Genome Analysis Center in the UK, Philippe Roby from PerkinElmer, and Dwarkesh Parihar from a wide variety of seed company and agricultural experience in India. Our format for today's webinar is going to be that we'll have all three presenters go through their content. We've set aside a question and answer period at the end of the session. For those in our remote audience, the way to enter a question is to use the blue question and answer box that's in the lower left-hand corner of your environment. Enter your questions there, and then after the three presentations, I'll be happy to relay those questions to our presenters. So our first presenter is Gawain Bennett. As I mentioned, Gawain is an automation specialist with the Genome Analysis Center. He's been working extensively in the library construction group, where he works on adapting and modifying NGS protocols for use with high throughput automation. Philippe Roby is a PerkinElmer principal scientist working with the alpha technology at PerkinElmer, and Dwarkesh has had extensive experience in crop science and agriculture in India, and will be our third speaker, presenting from a background of various experience in biotechnology applications as they apply to crop science. Without further ado, I'm going to pass the control over to our first speaker, Gawain Bennett. And Gawain, it's your show.
Hello. As Rick said, my name is Gawain Bennett. I work for the Genome Analysis Center, or TGAC as it's known as. I'm an automation specialist, and I basically look at automating and implementing new protocols and integrating them into existing pipelines, and develop new and custom automation for various of the sort of research pipelines that we are putting together at TGAC. Okay, I'm going to talk today about a low input and inexpensive sequencing pipeline we put together to work with the wheat genome. And before I do, I'll just give you a little bit of TGAC background. So TGAC is a relatively young institute. We're about 5 years old. We just celebrated our fifth birthday. We're a BBSRC funded institute, and we're located on Norwich Research Park in Norfolk in the UK. And we're a kind of technology and innovative driven institute focusing on health and security and animal, plant and microbial research. As you can see from this next slide, we're not particularly fussy what kind of organism we work with. We've worked with many things over the years, including ash dieback and the mite that's been infecting the European pollen bees, and we've worked with naked mole rat, dog, a mouse, and of course wheat and many other crops. Before I go into the main part of my talk, I'll just give you a quick tour of our labs. We have 5 Illumina HiSeqs. One is a HiSeq 2000, and 4 are HiSeq 2500s. 2 of which have the high capacity terabyte capacity run module. We also have some Illumina MySeqs. We have a PacBio. We're also on the MinION Access program, and we've also recently taken charge of a BioNano Iris, which is a mapping system. Now onto the sort of more interesting machines. I work in library construction. I work with liquid handling machines, and in our library construction lab, we have a whole suite of PerkinElmer machines. Initially, I wasn't there when the decision was made to go with PerkinElmer for our library construction, but it came down to the fact that PerkinElmer had a wider portfolio of methods that we wanted to run. And with hindsight, I don't think we could have got as far as we have if we hadn't bought these machines. They're very versatile for what we want to do. We have 3 cyclones. These are liquid handling machines. These handle our routine library construction. Anyone can submit any sample in any state to us, whether it's for sample QC or library construction or sequencing or primary analysis or even further analysis. You can do any one of those. I also develop on these, or me and my group, or the group that I'm in, we develop new and bespoke methods on these. We also have in our QC lab a Zephyr and a Twister. The Zephyr was originally brought in to do sort of various cleanups, but it's kind of become, does lots of sort of things in the post-PCR area, like amplicon normalization and PCR cleanup. As I said, GX and plate reader setup. It's quite a useful machine. And also we have various other QC machines. We have a GX and a LabChip XT. We also have an NGS Express for the more smaller scale stuff. Sometimes you've got a handful of libraries that you don't want to set up a huge cyclone to do, and there isn't anyone available to do them, so we can run these on our NGS Express. And we have sort of various other things that we have planned for that. So on with the main part of my talk, sequencing the wheat genome. So why sequence wheat? The International Wheat Sequencing Consortium got together several years ago now to try and generate a high-quality reference for the bred wheat genome. It's the third most important crop in the world after rice and maize, and one of its reference is not up to the same sort of quality as rice and maize, I believe, and it's thought that because food production has to increase by 50% by the year 2050 without the use of expanding land use or the use of fertilizers, and also that we thought that, or it's thought that if we can sequence the genome to a high quality, it will enable breeders to develop higher yielding varieties, new varieties that are better able to adapt to things like climate change and need sort of fewer fertilizers and pest control, etc. However, the problem with wheat is the wheat genome is complicated. It's very big. It's about 5 times the size of human, and bigger genomes tend to have more repetition. And wheat is further complicated by the fact that it's a hexaploid, and it has essentially three subgenomes that are homologous to each other. Each genome has 7 chromosomes, and the genome is not only, it's a hexaploid, but it's also it's very highly repetitive and is very GC rich. So the International Wheat Sequencing Consortium simplified, or part of the labs that were involved in it, simplified the genome by separating or sorting the chromosomes into the individual chromosomes, the genomes into individual chromosomes, and sometimes into the chromosome arms. For us to work at them at TGAC, we needed to simplify them further. Basically, if you have a chromosome, you have the same problem that you have with the whole genome, it's just it's a lot smaller. So we needed to simplify it further, and how we do this is using an old sort of tried and tested technology from Sanger sequencing days. I don't know if everyone's old enough to remember what a BAC is. A BAC is a bacterial artificial chromosome. Essentially, you take your genome of interest, or your gene of interest, you cut it up into little bits, you insert each one of those little bits into an E. coli, grow it up overnight, and you have many copies of your gene to prep and sequence. However, just this process alone will give you a lot of redundancy. You'll have many, many copies of your gene of interest. So the process is then sent away, or the library is then sent away for further processing to create a minimal tile path. This is just a minimum number of BACs that is required to get through a particular sequence. For wheat 3DL, which is the part of the genome that TGAC was working with at this time, it is greater than 6,000 clones. That's 6,000 libraries you have to make. And just to give you a bit of perspective of how big this task is, this is the map of the chromosomes that have been given out around the world. And you can see here that I'm pointing to 3DL, which is the long arm of the third chromosome on the D genome. So there's a lot of work to be done to get a good sequence. Okay, so you've got your library, you've got your minimal tile path in bacterial cultures. You need to extract the DNA. So we have at TGAC, we're lucky enough to have 2 robotic platforms. We also have a Beckman Coulter machine, which we use a modified cosmic prep to extract the DNA from a BAC library, 384 samples at a time. And the process has been, it's been developed to the extent that it gives very high quality BAC DNA at the end of it. However, BAC inserts are varying in sizes. Some of the bacteria grow slower than others, so the yield can be very, very variable. And there is no way we could normalize this to prepare for library construction, because it would take so much time and effort. So we kind of have to go with this sort of spray of concentrations, and also there'll be different sizes as well in there. So our method is a sort of quick and dirty method, and we're going for, I guess, quantity more than quality. So the library construction is a modified Nextira protocol. When we first started developing this method, it was Nextira was owned by Epicenter, and we optimized it for that. But then Illumina bought it out and took it over and modified the protocol. So we've changed it, and we've changed it to the extent that the only original part of the Illumina Nextira protocol that we use is the tagmentation enzyme. Every other reagent has been replaced. And we also, because we knew there would be much scope for multiplexing, you've got to remember a BAC, no matter how big it is and complicated it is, it's much smaller than a whole genome. Our BACs range from about 75 kb to about 250 kb. So the scope for multiplexing is great. Darren Heavens, the senior research assistant who was, I guess, the driving force behind this particular pipeline, he designed 48 forward and 48 reverse dual indexing barcodes. So we had a capacity to pool 2,304 samples. However, we started off with 96 samples, and to get the pipeline to 96plex, sorry, to get the pipeline going, we quickly moved up to 384. But before I go further, I just want to divert into a little bit of automation, because this is the bit that I'm really interested in, the bit that I do. Just working with these kinds of libraries is a phenomenal job in logistics, and this is one of the things that stuck us for a long time, is the bead-based cleanup for the post-tagmentation, which essentially, so in a 384 well plate, there was 20 microliters sample, 20 microliters of beads. You displaced about 30 of it if your tip went to the bottom, but these had to be perfectly mixed and with no air bubbles. If any air bubbles got in there, they would kill the BAC. So this picture here was the first time I got it right, I always stick this picture in any talk about the BACs, because it's, yeah, this was, for me, breaking the back of most of the automation was this bit. So we have a pool of libraries, and we then use a pippin prep, a Sage pippin prep to size select them, and they're cleaned up at that point as a pool and submitted to the sequencer for sequencing. So this is the results from the very first plate that we ran. You can see that even we were sort of overwhelmed with the number of reads we got with each BAC, and you can see that the coverage is quite a lot. So we realized that there was much more scope than we realized for multiplexing, and we were trying to aiming for 30x coverage. This sort of gave us a buffer for any sort of small amounts of contamination and what have you, and so we could expand the pooling to even further to 2,306, or 6 times 384 libraries. I can't remember exactly the number off the top of my head. So the assemblies look like this. So that this is one of the very first single BAC assemblies that we got together, and you can see where all the BACs are aligning to each other, but you can still see there's a very high, what we believe to be a high GC area, or there's areas that the sequencer can't seem to get through. So, and like I say, it was at this point here that I left the program. I'm an automation person, and as soon as the automation was finished, I moved on to pastures new to do other high throughput methods. But Darren Heavens continued on. He wanted to improve the sequence even further. He wanted to put some scaffold sequence in there. So he chose a, oh, sorry, I've skipped a slide. Sorry, this is just some statistics from the assembly. So at this stage here, the 6,144 clones that we were working with, the average contig was less than 18 and greater than, the N50 was greater than 13 kb. Okay, it was this point I left. Darren went on to work on using a long-made pair protocol to try and put some scaffold in. If you don't know what a long-made pair is, basically you circularize a big fragment of DNA, and where it joins at one end, you take that out, that's your mate, and you know that between those two sequences it's of a given length, and here you can see it's 6 to 8 kb. Darren's strategy was very interesting in that he just took the DNA of the straight from the BAC prep and pooled it all together and did the long-mate pair in a single reaction. And the theory is that each mate would only map to one BAC, and you just, I guess, what's the word, blast it back to the original BAC. And as you can see here, again, there was much scope for multiplexing, and he's got it to the point where he can do 12 times 3, 4 well plates. That's 300, so that's 12 long-mate pair reactions pulled together down a high-seq lane. Okay, so the long-mate pair data vastly improved what we had. You can see here there's a number of graphs where you can see that the number of contigs has dropped, and the assembly size has got bigger. And I've put a note in here, the larger assembly sizes show increased percentage of ends, and we believe that's because of the variation between the two mates. I don't know if we've quite got to the bottom of that perhaps some of those ends don't actually exist. Okay, to show the sort of final improvements, the average sort of scaffold contig number has gone down to less than 7, and the average scaffold N50 has gone up to 90 kb. And this is kind of where the 3DL project is at the moment. All the library construction has finished, and for the moment we may have to go back and do some more depending on what needs to be done. As you can see, it's sort of in pretty good condition. There's a quarter of it assembles to a single contig, and the big spike at 11, that should say 11 plus number of contigs per BAC. And now it's with the bioinformaticians who just have to stitch all this data together and try and make a full 3DL sequence. And that's pretty much my story. So the future for the BAC pipeline is that Darren's going to publish this work, and he's continuing to tweak it, and he's working on other problem chromosomes, Barley2H and the 0H, which is the unclassified chromosomes, Lolium, which is another grass, and he's looking to increase the capacity to 4,608 samples per lane. And he's also looking at running the long-mate pair pools down the same lane. So, yeah, I think that's pretty amazing. For me and the robots and I have moved on to a pre-capture pooling exome capture pipeline. Again, greatly multiplexed, also to do with wheat, but a completely different project. I'm also looking to do RNA-seq in a day. I've had some pretty good things about Lexogen and a high-throughput PacBio process. So that just leaves me to thank a few people. I'd like to thank Matt Clark, who was the principal investigator in this project, and he's also the head of plant and microbial genomics. Darren Heavens, senior research assistant and the driving force behind, the development of the protocol, and the amount of hours we sat talking about how high tips should be. Leah Clissold, my long-suffering line manager. James and Heather used to run the pipeline with us. It used to take 4 people when it was in full swing, but we've sort of tweaked it enough to the extent that only one person needs to run it now. I'd also like to thank Dipali Vasoya, who one point was the only bioinformatician on this project, and she was basically given a lifetime's worth of work with the amount of data we were generating until Sarah Ayling's group took over. Durania helped me with some of the graphs, and I'd like to thank Paul and Nancy from PAC and Elma for inviting me here to talk today, and last but not least, Tracy, whose kindness and patience has been immense over the last few weeks. Thank you. So I'm now handing over to Philippe.
What I'm going to describe to you today is a new technological approach that will allow us to actually generate immunoassays for detection of in plants. So why do we need immunoassays for plants? Actually, we need them all over the place. If the goal is actually to create a new plant, like a transgenic plant, you start with actually the gene discovery and protein expression. So you will have to measure that by using an immunoassay. Then you'll have to create the plant in question. So you'll have to do immunoassays at every step to follow it to prove that it works. Then you would have to go to regulatory, so you have to prove to the FDA it actually works. Then in production, you'll have to prove to your manufacturing department that it actually works. And finally, you'll have to prove to the customer that it actually works. So the gold standard generally used for immunoassays is ELISA. It is a well-established technology, but it has some handicaps, especially if you want to go into mass screening or mass testing. So I will present a new technology called AlphaLISA, which is an ELISA replacement technology that shows some advantages at this level. So the first part will be to describe what is AlphaLISA and how does it work. And in the second part, I will present examples of genetically modified plants in which we are able to detect the introduced proteins. So what is alpha? At the basis, it is a bead-based technology. You have two beads, the blue donor bead and the red accept bead. These beads can be brought together by molecular interaction between partners that are fixed on them. The goal here, for example, would be the detection of an antigen by partners A and B. When they are brought together, upon excitation of the donor bead with infrared laser, chemicals into the donor bead will generate a quantum state of oxygen called singlet oxygen. This will then diffuse to about 200 nanometers to penetrate the accept bead. In the accept bead, there are chemicals that will react with singlet oxygen to actually generate light. And that light is the output you are going to measure. So two important things to note here. First of all, this is an amplification technique. For each bead, there are thousands of molecules of singlet oxygen that can be generated, and there are thousands of chemicals in a single acceptor bead that can receive the singlet oxygen and generate photons. So you can get many photons from one interaction. The second thing to note is that the singlet oxygen can diffuse up to 200 nanometers. While this may look small, this allows alpha to measure about any biological entity you can want to detect, from very small molecules like amino acid or nucleotides up to very large proteins such as IgMs or even viral particles. So how is AlphaLISA actually an ELISA replacement technology? It is actually very similar. On the left, what you have is the classical ELISA system that will be sought to detect known analytes. At the bottom of the plate in green, you have a capture antibody that is bound to the plate. This will bind the analyte, that green squarish thing. It will then be detected by the blue antibody, which is called a detection antibody. This antibody can be detected by other moieties. In that case, it is biotinylated and recognized by sortavidin. The sortavidin is labeled to an enzyme that will convert the substrate present in the ELISA mix to a color molecule. So you actually measure the color. This is where the amplification happens, because this enzyme can convert several molecules of substrate. The conversion is actually straightforward. The IgG used for capturing, instead of being bound to a plate, is bound to the acceptor bead. The analyte is recognized by that antibody, which is then recognized by the blue detection antibodies. And instead of having sortavidin labeled with an enzyme, you have the donor bead, which is also labeled with sortavidin. So then the singlet oxygen can go between the two beads and generate a signal. The amplification coming, like I described before, from the fact that you can have many photons generated by one bead pair. On this slide, you show that creating the bead, especially the acceptor bead, is actually quite simple. You don't need complex chemical interaction or activation of the antibody or anything. It's just a mix and wait technology. You just put the antibody along with the acceptor bead and a mild reducing agent to obtain an amine bond between the antibody and the bead. This allows you easy, reproducible, a mild treatment that doesn't require much antibodies, but has also the advantage of giving you a stable covalent bond that will resist any kind of biological matrix you can put in there. So there's no risk, for example, of the antibody leaching out of the plate if I were to use a large amount of detergent. So there are actually two formats then you can use to do your ELISA. The typical one on top that has been previously described is called the sandwich assay, which is a signal generation assay. The more analyte you have, the more the beads are brought together, and the more signal you actually get. Unfortunately, in some cases, you may want to detect a very small molecule. So in that case, the molecule is too small to having two antibodies be able to bind it at the same time. So what you do is actually you have a labeled probe, which is analyte that can bind directly to the donor bead and is recognized by the acceptor bead. So if you have analyte in your sample, it will compete that interaction. So you have a signal decrease assay. These can be useful to detect things like amino acid or nucleotides, but when you're detecting proteins in 99% of the case, it will be the sandwich assay which will be used. So now to show you the protocol compared to an ELISA, as you can see, it's extremely simple. You just add your analyte into the wells of a microplate. You add your acceptor bead and your biotinylated antibody, or in ELISA terms, your capture detection antibody. You incubate for about 1 hour in most assays. You have your donor beads. You incubate for about 30 minutes in most assays. You then excite and read the signal. As you can see here, this assay is very fast. Including pipetting time and creating your dilution, it will take about 2 hours. And as you can see also, it is an homogeneous assay. So there is no wash step in any portion of the assay. So you gain on time, and I also discuss a little later, you also ease up your optimization. So now that we have presented alpha, we're going to see it at work in plant assays. So the goal here was actually to create an assay that could detect transgenic cotton. These cotton seeds were modified by the introduction of a gene from Bacillus thuringiensis, which is a protein that actually kills insects that will try to eat the seeds or the other portion of the plant. There are several variants. Two here are mentioned, Cry1AC and Cry2A, that will be the target of our assays, but there are several other variants. So the current assay is actually a 96-well plate ELISA that uses one seed to do the assay. It also provides a qualitative answer. So there is a protein present or there's no protein present. So the request here was actually to replace the ELISA with AlphaLISA for several reasons. First of all, the goal was to reduce the price of the assay, was to introduce the potential of actually multiplexing at least two of the proteins, and increase the throughput by increasing the use of automation and actually moving to smaller well plates, like 384 well plates, so the throughput can be massively improved. So we actually took a look at it and decided this would be feasible. So then we started to work on it. Top left, you have the actual assay itself, which is a typical sandwich assay between an acceptabile and a bivalent antibody. In that case, the assay described here would be Cry2A. So what we did was actually use commercial antibodies against the assay. So on the right, you have a graph when we tested various antibody pairs, detection and capture antibodies, by either labeling the antibody to beads or biotinylating it and trying to find a good pair. We actually found two possibilities. One was to use the same antibody on both sides. The other one was a mixed pair of antibody. As you can see, the third antibody didn't give any signal and was rejected. So for technical reason, we prefer to have two different antibodies. So we used two connection antibodies to actually generate the assay. The second step, lower left, is actually optimization of the assay. There are ways to tweak the protocol to try to improve sensitivity and signal output of the assay. So what we did here, if you remember the protocol, instead of mixing the two antibodies and the analyte, we mixed only one antibody with the analyte, incubated, then we added the second antibody, then we added the donor beads. The results are in blue. We call it the three-step assay. And as you can see, if we use that assay, the signal is significantly increased, and the sensitivity is increased by 3 times. So these assays were done with recombinant protein, but what they show us is that we can actually make an assay. We do cover the need for a yes or no. We can have a very nice assay window showing the presence of the protein. And we went a step even further, because by using the standard curve we show here, we can actually measure the amount of protein in each seed. So the next step was actually to see if we can do it in seeds. So we used a very easy seed extract protocol, which is simply grind one seed, or about 90 milligrams of seed, are treated with an extraction buffer for an overnight, and then centrifuge to remove seed particles and test your protein. As you can see, we have very neat assays. The high bars are actually the seed that were transfected with Cry2A. The very small bars on the left are the ones that were not treated with Cry2A, we call the wild type seeds. So you can see we have a very good difference between the actual treated seed and the wild type seeds. The tree graph represents 3 different types of extraction buffer, confirming that very soft and very easy extraction buffers can actually be used. The lower left is the most interesting because that extraction buffer is also the assay buffer we use for AlphaLISA. So you don't need to introduce special buffers or special extraction condition. So now that we had a functional assay, we will ask if we could multiplex this assay. So we decided to test it. The multiplex process is actually to introduce more and more acceptor beads into the assays. Here you have an example that we can do with our instrumentation and technology to do a duplex. You have your blue donor bead in the center. You have 2 different biotinated antibodies, and you have 2 different acceptor beads. The combination here will be able to detect 2 different analytes. So in the same well, in the same assays, you can have 2 standard curve and 2 detection of 2 different analytes, allowing you to double your output and actually measure things like ratios of protein 1 versus protein 2. So we actually applied it to seeds. So the goal here was actually to detect 2 proteins, Cry1AB and Cry2A, in the same well, in the same assays. So here we actually tested 2 different new duplex beads. The EUTB is actually a combination of 545 and 615 nanometer emission. The EUSM is actually 615 and 642 nanometer experiments. So here you can see in the solid blue in both graphs, you have Cry2, which is detected by the 615 beads. The solid bars are the seeds that were transfected with Cry2. The open boxes were the ones that are the wild type. So as you can see, you get again a very nice assay window and a clear differentiation between the 2 with high sensitivity. In the red bars, you get Cry1 AB. Again, the solid markers indicate the transfected seed. The open markers indicate the wild type. So in a single assay, you were able to measure the 2 different components. At the bottom, you have a summary of the SB present. So you can see that in both cases, you can have assay windows of 50 and over that will give you a very clear response to the presence of the particles in question with high sensitivity, high selectivity, and high efficiency. So this was an example of one target. As you can see, this is a site that describes many sorts of transfected seeds that are available. So it shows that there are many targets where this technology could be applied. And this is actually a confirmation by us and other customers that the output and the efficiency of the technology is actually present. We can show, for example, that one technician can actually almost decuplate the amount of sample he can actually process. Sensitivity is higher. Sample volume is significantly lower, up to 20 times lower than the concentration needed in ELISA. And the data can actually be obtained in about 2 hours, which allow you to create at least 3 or 4 different assay runs per day. So in summary, Alpha has proven very applicable to plant protein technology. It is extremely versatile. It can detect various ranges of affinity with molecular weight. It can easily be miniaturized. All the assays that were shown to you were done in a 384 well plate. If you want to further miniaturize and go to 1536, all you have to do is divide every addition volume by 4 and simply add directly to the plate. They can use with complex biological matrices, such as plant extract in that case, seeds extract, or we also did it with leaves or other parts of the plant extracts. The conversion from ELISA to AlphaLISA is extremely straightforward, and the technology is cost-saving and very productive. The hands-on time is diminished, especially because there's no need for washing the plates or things like that. It consumes very low amounts of antibody and also very low amount of sample. And we also show it was capable of multiplexing. So I'd like to thank the people that actually took care of this project and generated these very interesting data. Application specialist Lindsay Nelson, senior application specialist Steve Ert, and the manager of this assay, Vincent Duprier. So now I'm going to send the speaker to our moderator, Rick Bunch, who will take over at this step.
Good morning, ladies and gentlemen. This is Dwarkesh Parihar. I'll be talking about application of high-throughput genotyping for marker-assisted selection in plant breeding. This is something which is quite useful in almost all types of trait introgression or any gene linked to a marker and introgression of such a gene. As I mentioned earlier, that genotyping forms a very important component in marker-assisted selection and accelerated backcrossing when we are going for transfer of GM or non-GM trait from the donor genotype to any recurrent parent or parents of the existing hybrids. And this is done in many different ways, starting from using SSR markers, and before that it was RFLPs and so on. In this slide, this is a typical backcross program for trait introgression, which is used in majority of the trait introgression programs, whether in seed industry or in public sector institutes, where a gene, here in this particular case, it's a dominant gene, needs to be transferred to a recipient parent, and you make a cross, get F1, carry out foreground selection, and then select the plants which have gene. Of course, it will be in the heterozygous condition. Cross it again with the recipient parent or recurrent parent, and you go on till BC3, and after that this progeny can be selved to obtain BC3F2, where using foreground selection one can select for the plants having the gene in homozygous condition and so on. During this whole process, if we see foreground selection is carried out to select for the presence of gene or trait of interest, and background selection is carried out to select plants carrying higher genomic component of the recurrent parent. And this is where it becomes very critical to select plants in each generation, the plants having a highest genomic component based on genotyping, which can be carried out by SSR based markers now or SNP based markers. And this really accelerates the whole process of backcrossing. So conventionally, if we see this slide, that conventionally if we compare traditional backcross versus marker-assisted accelerated backcrossing program, in the traditional backcross, usually up to backcross 9 generation carried on, and still we see it's not really complete recovery of the recurrent parent genome. On the other hand, if we are using marker-assisted selection, in that case, by BC4, that is the backcross generation 4, we already have plants having almost 100% recurrent parent genome, and this is what is required for a successful backcross program where gene introgression is being carried out. And in terms of time, if we see the long cycle almost lasting more than 3 years if we are doing in a conventional way, and on the other hand, if it is done through marker-assisted selection, it takes the whole program can be converted in a shorter time, close to 2 years, which is almost half of the time which is required in the traditional method. And if we are using marker-assisted breeding for introgression of 1 to 2 genes, and if there are 1 to 2 recurrent parents where the genes are being transferred, then the conventional method, not really conventional method, but the method which was used, I would say 5 to 7 years back, involved carrying out foreground selection using PCR with specific primers for the gene or genes of interest, select the plants carrying the genes, and carry out background selection by genotyping using SSRs on such plants, and select the best plants from this generation. If we see this whole process, of course 7 to 8 years back this was okay, but if somebody has to carry out this process now, it looks quite lengthy and cumbersome, because the first thing is that reading of these gels, if there are just few gels, it is easy to read for the background selection. But when it comes to for the foreground selection, when it comes to background selection, it becomes even more difficult, because number of gels that one has to read is quite high. If on an average, if a scientist is using 60 SSR primers for genotyping, then it becomes almost 60 agarose gels to be read manually, and then compile the data and run it through the software to generate genetic diversity, and then they select the plants which are closest to recurrent parent. So this is that is the whole process which I have explained in this slide, where if we are carrying out introgression of 1 to 2 genes in 1 to 2 recurrent parents. So if we look at one gene for foreground selection, and if we carry out PCR based selection, then out of 100, 50% of the plants, they turn out positive for the gene. Then when we move on to the background selection, on an average, if we use 60 SSR markers for background selection, and in 2 segregating population, then the numbers almost become 100 plants, and then it is tested with 60 SSR primers, then it becomes 6,000 PCR assays. Now we see the numbers are multiplying, and To run this many PCR reactions, we need at least 24 PCR. If we are running 24 PCR reactions per agarose gel, it becomes almost 250 gels for data analysis, and the output of that data will result in selection of plants. So manually reading such a large number of gels is cumbersome, and it leads to certain errors during manual data calculations. So overall, the handling of samples in this way is messy because at times we end up with agarose gels which are either smiling or frowning, and that results in error in reading that kind of agarose gels. When it comes to data analysis, again, it is time consuming, and when it is done manually, it's prone to errors. The other important part in a BAC cross program is the isolation of DNA, doing the PCR, getting the results, and the flowering time of that crop. So usually it happens that if we have a lengthy, cumbersome process, then it becomes difficult to match the flowering time of crops, and then at times there are delays in carrying out crossing. The other problem associated with this is that few populations can be handled. Thus, it severely restricts utility of marker-assisted selection when we are working with large-scale application. Hence, what we need really is a rapid way of genotyping of large number of samples. Several segregating populations can be handled simultaneously. That is what is the demand of a successful backcrossing program, where marker-assisted selection actually accelerates the whole process and makes the whole process quite easy to handle. And when we talk about the rapid genotyping, again, there are several options available compared to agarose gel. There is a possibility of using systems which are more automated, taking less time, so the whole process, again, for rapid genotyping would involve isolation of DNA from large number of samples, then a good appropriate PCR setup to generate amplicons for analysis, and followed by setup for rapid, reproducible, and automated analysis of amplicons, which in the conventional way is agarose gel, and followed by appropriate software tools for large-scale data acquisition, analysis, and presentation of results in a simple, readable form, so that the researchers at the field level can read it easily, understand it easily, and carry out selection of plants for backcrossing. And one of the simple method which we have used in our lab to speed up this whole process of marker-assisted selection in a practical breeding program in a seed industry. I work for hybrid seed industry, where marker-assisted selection is quite common for different types of introgression programs, whether it is introgression of transgenes or introgression of genes linked to a marker, stacking of genes linked to markers. So all these things we do almost on daily basis, and there we found that making a shift from agarose gel electrophoresis to a microfluidic chip capillary electrophoresis really helped in speeding up the whole process and carrying out the analysis in a very timely manner. So eventually what we learned from this was that it is critical to have a nice, very efficient system at the stage of genotyping. In this and also the system which we used, which is based on microfluidic chip capillary electrophoresis, which is essentially a miniaturization of what we call sort of agarose gel, but everything is miniaturized and automated. So, and the instrument and software, they control the movement of fluids through these optical channels, and with the help of laser, the movement of DNA bands is detected and recorded. So essentially this system takes over the electrophoresis and data acquisition part. And the system, with this kind of system, one can use a 96 well PCR plate, and if we want to shift to higher throughput, then definitely it is amenable to a 384 well plate format also. Only difference is that in this kind of situation we need PCR machines whose block is also in 380 well format. Once the PCR reactions are run through this kind of microfluidic system, and it is fairly simple. After the PCR reaction, you just take your plate, take off the cover and put it on the stand, and just switch on the software. And if you are interested, you can stand in front of the monitor and see what is happening. But after a couple of runs, then you realize that it works on its own. There is no need to stand there and watch. So essentially, you put the things there and just go and do something else, whatever you have to do, whether data analysis from previous experiments or so. So in this slide, this is a representative electrophotogram from one of the population where it is showing polymorphic allele, and as you can see, the size difference between this allele and this one. This is a larger allele compared to this one, so this is a polymorphic allele which can be easily detected with this kind of system. Otherwise, if I just compare it with the agarose shell, then probably all these three alleles may be difficult to differentiate on an agarose shell compared to a microfluidic system, and this kind of results are pretty easy to read and analyze. So here is this example of a polymorphic allele. If we move on to the next slide, this is an example of monomorphic allele. So there is no polymorphism between these two samples. And the third example I have taken is a missing allele. So you see here we have three alleles, and in this sample we have only one allele present. So the best part of this system, the data, the data acquisition and its analysis is so good that even the minor differences which would be otherwise missed in an agarose shell can easily be detected. Thus, it results in a higher number of data points per PCR primer that we are using for genotyping, and it is easy to analyze the data which is coming out of this kind of DNA analysis. Apart from these electropherograms, the system also generates virtual gels, which can be easily used to compare the results which are there on the electropherogram versus a virtual gel image. Actually, when we started using this system, we ran a couple of PCR reactions where the samples were run on the microfluidic system and also on the agarose shell, and at times we found that the level of detection in case of microfluidic system, it can detect DNA bands in nanogram quantities, which at times becomes difficult when we are using an agarose shell. So overall, the results generated by such a system are I mean, we are quite happy, and initially we were quite impressed because it made our life easy. Coming on to the data part, that the system acquires data from the DNA samples being analyzed, and this can be easily exported from the, of course, we are using LCGX. So from there, the data can be exported to Excel sheet just by click of one button. And from here, you can easily see that what is happening, that what kind of alleles are there. Like if this is an example where we have these 4 alleles which are there, and they are not showing any polymorphism. And also it shows the concentration of DNA in the virtual gel. So you can see that it can detect almost 1.8 nanogram, even less than 1 nanogram very easily. So once this data is transferred to Excel sheet, then by using some tricks, these can be converted into a binary table where we have our recurrent parent, the donor parent, and the alleles which are detected by the system. And you fill in this sheet, transfer to any software. We use NTSys PC for further analysis, and select best plants for back crossing in the next generation. This whole process is again a very simple one. One person can do, I don't know, maybe 4 to 5 segregating population in a day. So analysis of the data coming from 4 to 5 populations in a day. And eventually we end up with a dendrogram. We use dendrogram, but there can be several other outputs. The data can be used to generate several different types of outputs. We found that for the people who are working in the field, for them this is quite easy to understand that here is my recurrent parent, and these 3 plants are the ones which are closest to my recurrent parent. So these plants are the ones which the person at the field level will select and carry out back crossing with the recurrent parent. And this whole process is very convenient, because time taken to do the whole analysis is now short, and very easily we can match the flowering time of different populations and do things in time. So coming to towards the end of my presentation on our shift from agarose gel to microfluidic system. So in terms of, if you do a comparison between agarose gel based genotyping and microfluidic chip based genotyping, so initial investment is low. On the other hand, for a system like this, it is moderate. Data points output is moderate versus high. This is because in agarose gel based genotyping system, we are not able to detect all the alleles, and so we end up with lesser data, and which may result in errors in the final analysis. Cost per data point is low in case of agarose, and it is moderately high, but it very well gets compensated by the efficiency of the whole system. Of course, if you look at the time, it takes a longer time compared to this kind of system. Manual intervention over here is minimal. You put the plate in the system and switch it on, and you can go and have a cup of coffee, or you can do something else, more important things to handle. And as I mentioned earlier, in terms of efficiency, so the cost per data point very well gets compensated by the high efficiency of this system, high overall efficiency. And also another very critical part is that when we are working with agarose gels, the gel images we have to store as data, and they take large amount of space in the server, versus very small size files generated by a microfluidic system. The one which we are using is LCGX, and where small size files can be, large number of such files can be stored without taking much space from the server. Of course, safety is important, and there no such chemical, the users are not exposed to any toxic chemicals like ethidium bromide, which are used in conventional agarose gel. And there's another important part is that this whole system can be automated. If there are large number of samples, then you can add more than one then one LCGX system, and a robot can be attached which can do transfer of the plates from PCR machine to the LCGX system, and then it becomes a fairly a simple automated system. And of course there are other options for automation which I'm not going to discuss now, but overall my experience with this system was quite good. I mean, definitely shifting from agarose gel to this kind of system makes a lot of difference, and I'm quite happy with this system. With this, I think I would like to say thanks for your patience and listening to my experience of having a shift from agarose gel to a microfluidic system, especially for genotyping, and of course it can be used for many other applications. Even it can be used for RNA analysis with certain changes. Even proteins can be analyzed. So it's a multi-utility system. Thank you very much.
And now open the question and answer session. The first question we have coming in, Gawain, is for you. For our remote audience, if you do have further questions, there are several that came through during the course of the webinar, but if you do have further questions, please feel free to post them in the question and answer box, which, at least on my environment, is the blue bar box in the lower left-hand side of your environment. So, Gawain, first question for you involves the scaffolding, and the question is, would the viewer is familiar with the PAC biosystem? Would the PAC biosystem fit your workflow for the scaffolding? And I'll pass control over to you to answer.
Okay. Is it? Oh, there's me again. I think in the very early days we considered the PAC bio, but I guess the throughput just wasn't great enough, and I think there was some PAC bio work done, but with the long mate pairs to build the scaffolding, you can generate so much data with so little reagent and sequencing capacity, and we went with the long mate pair. I mean, I think Darren's latest long mate pair run was 12 pools of 384. You just couldn't get that kind of data with the PAC bio, I don't believe. Has that answered your question? Oops, I have to pass it back to...
Thank you, Gawain. The second question is also for you, Gawain, and it's somewhat related. The question came in also regarding the scaffolding, and it's about the scaffold sequence. And the question is as follows. So, following your workflow, each scaffold sequence needs to be placed among the others. The question is, how do you plan to merge the 6K assemblies into a single assembly, and what tools are you using to do so?
I'm not really a bioinformatician. Oh, did you get all that? I'm not really a bioinformatician. I don't really know. In my head, what I would imagine is that you get all the BACs and stick them into a massive assembler and put them together, but I don't really know. And I spoke to the person who asked that question, and I'm going to look into it further. I can let you know the answer if you know. I'm so, what's the word, far removed from the bioinformatics that I really don't know how that would happen. I'm sorry. Pass full control. Yeah.
Okay, thank you, Gawain. The next question incoming is for you, Philippe. It's about the alpha technology in your presentation. The question is, in multiplex alpha, how can we assure the higher selectivity of the antibodies used to detect two different proteins and not getting interference or multiple attachment of the two proteins to one antibody?
Okay. To answer that question, I didn't show the data for simplicity, but every assay is first done individually. So I would generate one assay for protein 1 and one assay for protein 2, and then I would try to cross-match them, like use the assay for protein 1 against protein 2, and use the assay for protein 2 against protein 1. If I were to get any signal out of these cross-match assays, I would simply dismiss that as unworkable and try to select other antibodies that have more selectivity.
Okay, thank you, Philippe. The next question also is in regards to alpha. The question is the AlphaLISA quantitative? How many analytes have been tested successfully in your multiplex system?
So we're talking about two questions here. The first question is actually, yes, alpha is quantitative because as I showed on some of the earliest slides when I presented the plant results, we can actually see that we are basing our result on the standard curve that was done in the same matrix as for the assay. So you can easily plug the signal you get from your seeds on that standard curve and actually measure the exact amount of protein present in your seed sample. The second question, how many duplexing assays did we do? We did several of them. I would say we have concrete data for about half a dozen, but we are working on more every day now, and we should have a more complex list. The ones we did involved several different domains, including plants, animals, biotherapeutics, and pure biochemical assays.
Okay, Philippe, thank you very much. The next question is back to Gawain, and it's about some cost figures for your workflow. And the question is, what is the approximate cost of each BAC from your workflow?
Okay, so currently the cost is £5 per BAC from BAC to sequence. We did have something in mind to get to £4 to do that, because we originally were a Sanger sequencing lab here, and we used to sequence back-end sequence, just the ends, for £4. And I don't know why that's become the sort of target, but it was about £7, £8, and it's come down to now about £5 per BAC. So it's quite cheap.
Thank you, Gawain. And in similar fashion, Philippe, there's a cost question for alpha, and the question is, I guess, twofold. Are the costs per sample for alpha lower than standard ELISA? And two, what steps allow, if it's lower, the cost to be lower with alpha than ELISA?
Okay, to answer the first part of the question is yes, the costs are usually lower than for a regular ELISA. The cost reduction actually came from several portions of the technology. First of all, you save on time by neglecting the needs for things like blocking steps and wash assays. So you save on the FTE portion of your assay. You also save on issues like your plastics, like you need much less tips. For example, if you don't do wash assays, and since you can miniaturize, you don't need as many plastic plates. Finally, the amount of antibody needed to conjugate your acceptabines and do your actual assay by adding the biotinylated antibody are extremely low. We're talking amounts here ranging in the single nanomolar range, which would probably indicate in your well a couple of picograms of antibody. So with a regular sample of antibody like 100 micrograms, you can run literally thousands and thousands of assays, which is not necessarily the case with ELISA.
And the next question is right back to you, Philippe. The question is, how will you relativize your data once you get beyond the AlphaLISA, and how much background do you get compared to a normal ELISA?
So the background issue is actually something you can control. Background is actually extremely low because the chances of two beads crossing each other randomly compared to the volume of an assay well is extremely small. So there's also very little risk of anything sticking to the beads. In some cases, you may have to optimize the assay to guarantee that, but usually it is not really an issue.
Okay, another question that pertains to the alpha technology, and it's in regards to duplexes, Philippe. The question being, is there a capacity for 2 duplexes, and how does this work in the alpha system if there indeed is capacity for multiple duplexes?
Okay, we haven't tested really so far having more than 1 duplex in a single well. We have done duplexing in a single well. If you want to add even more combinations, then you would probably need to, like a triplex, then this would probably mean assay optimization and special protocol for reading, but it is something we can easily work out. There's no actual technical obstacle to doing it. It would just need a slightly different protocol than the one you use for simple end duplexing.
Okay, thank you very much. I want to again extend a hearty thanks to our speakers today, Philippe Roby and Gawain Bennett. I hope you've enjoyed this session as a remote audience as much as we enjoyed putting it on, and also hope you look for future content from LabRoots and PerkinElmer. So we're at the top of the hour. We're going to close our session now. Thank you again all for attending, and please look for future content in this webinar format. Thanks. We'll be signing off now.